Yeah, thank you so much for such a kind introduction. And it's really my honor and privilege to be in this room, I think sharing our work with the experts from, I think, industry, academia. What I'm going to talk to you, I think, a little bit about the whole field, okay, spatially resolved omics profiling by sequencing. But largely our own technologies developed over the past couple of years in my laboratory. And so this is my disclaimer. So when I kind of switched my career back to 2006 from completely different field, like a physical science, energy, battery stuff, to biomedical research when I started as postdoc at Caltech, I was tasked to look into the complexity of the tumor microenvironment. And that time I was very naive about biology, but I realized two things, right? It is complex. And also first time I realized tumor is not just a bag of tumor cells. Actually, maybe that does not surprise you guys, but that really surprised me that time. And I think, oh, we really need a single cell resolution tool to really kind of understand the complexity and the heterogeneity of the tumor microenvironment. And number two, if you really want to pinpoint what those cells are and how they do their job in relation to other cells to kind of collectively shape the tumor microenvironment, you need a spatially resolved technology to map down the cells. So that time I was interested in how kind of inflammatory cells and the tumor cells, how they talk to each other. The analogy I used often is I very much like how I'm presenting here. I can speak English, maybe other languages, but unfortunately cells don't speak English. Cells don't speak other languages either. They speak something called cytokines, chemokines, growth factors. That's how they kind of go through their cell-cell communication by the paracrine signaling. So, and I ask myself, can we develop technology to map down or to kind of decipher the language and the cells speak to each other, then you know what they're going to do, right? So I think that's the best way to figure out what they're going to do by asking them to speak. So that was really my very first research, and I continued to kind of push the boundary to develop this single-cell protein cytokine secretome analysis technologies, but I'm not going to go into too much detail. Also, as alluded at the beginning, that has been commercialized by a company, and now it's commercially available at the Bruker. And since then, we continue to look into single cells. Okay, often people kind of forget we are still working on single cells in my lab. So one example is the CAR T cells. Turns out whether or not you really understand the complete functional spectrum once they become activated in an antigen-specific stimulation manner, it's still not fully understood. It's hard to do. And we utilize a range of technologies, including the one we developed, to characterize the single-cell cytokine-specific antigen stimulation response. And we characterize that from all the patients, the CAR T cells isolated from all the patients in the first two CAR T clinical trial conducted by Carl June, Steve Grubb at UPenn. We discovered something extremely exciting. We never thought about it. I think no one thought about it. Stay tuned. I hope that paper will come out in a couple of months. But anyhow, I think until now, I still have not talked about the second aspect I mentioned at the very beginning. Tumor microenvironment, single cell resolution is required, but ideally you want to look at that directly in the tissue context, right? So people realized that challenge a long time ago, but still kind of quite limited by the tools available to do that. About 10 years ago, this whole field started to emerge called spatial biology. Back to that time, I think when I co-organized a seminar series with Ahmed Koska, I believe he's speaking today as well. And we were debating what's the title of that seminar series. And I told Ahmed we should call it Spatial Omics. It's not spatial genomics, spatial transcriptomics. I never thought I'm going to develop another spatial transcriptome technology. Let's make the family as broad as possible. That's the community eventually that's going to empower scientific discovery. We need to work together. We need to join the force. So about 10 years ago, I think our microscopy or imaging experts really pushed the boundary by developing the extremely high plex, either protein imaging or fish. I think two technologies or two different high throughput fish. Let me see where the pointer. Yeah, it is. Eventually, they were able to even demonstrate whole genome-wide or transcriptome-wide gene expression imaging using single molecule fish. And for the protein spatial analysis, there are several different approaches to use. For example, the mass tag to image the protein expression using mass spec and then the specific masked antibody conjugate mass probes to image the presence of the protein directly in the tissue. You can also do cyclic immunostaining and image over multiple rounds, and eventually can get an extremely high plex. So I think the whole proteome-wide imaging probably is still challenging, but I would say about 10 years ago, if you do immunofluorescence staining, if you can look at 5 markers, people are really jealous. You are really the best in the world. I would like to work with you. But nowadays, I think a paper that was published just probably 2 weeks ago by Akoya Bioscience and a group of scientists in Australia that demonstrate you can actually image up to 101. What I heard is 103, but eventually they were a little bit conservative. I think 2 of those were maybe not image over all the samples they put in that paper, so they just try to claim only 101, but it's still stunning, right? So now you can image more than 100 proteins simultaneously from the same sample. So that's the world record. So nowadays, about 5 years ago, 5 to kind of 7 years ago, people realized you can also use high throughput NGS sequencing to map down, in particular, protein gene expression, but you can also use the high throughput NGS sequencing to map out other molecular information. That has been kind of our main interest. So the landmark paper came out 2016 from Professor Joachim Lamberg's laboratory in Stockholm, Sweden. He reported the use of DNA microarray to capture messenger RNAs directly released from the permeabilized tissue section you place on top of the tissue microarray glass slide. And the DNA microarray spots, each of those has unique sequence. You can call it a spatial address code or spatial barcode. Each spot has the spatial address code, but also has something like a poly-T, oligo-DT that can capture messenger RNA. And afterwards, you do on-glass slide cDNA synthesis. You're going to incorporate those spatial address code into your cDNA sequence. So that's how come you can generate sort of spatially encoded or spatially sort of identifiable cDNA molecules from just one tissue sample, and you can resolve that later once you get all the cDNA collected and perform unsupervised, high throughput NGS sequencing. You can read where that spatial address code is and what that gene is. That's the fundamental principle. So I think many of you probably know that that has been commercialized by Genomics as the Vision platform, and since then, actually many other technologies were kind of reported to show this has even greater potential to achieve single cell or even subcellular resolution spatial whole transcriptome sequencing by reducing the size of the spots or size of those pixels. In particular, 3 technologies are truly stunning. It's called a stereosec and a 6-scope and a pixelsec, and the spot size could be as small as 0.5 micrometer. But they all follow the same fundamental principle Professor Joachim Lamberg coined, called a barcode solid surface for messenger RNA capture and a spatial transcriptome sequencing. All right, so what we came up with a couple of years ago was kind of total opposite. We don't capture messenger RNA on the glass slide. Instead, we were thinking, can we send those spatial address codes into the tissue to barcode whatever you want to barcode? Turns out that's extremely versatile. We can barcode transcriptome, but we can also barcode many other things, including proteins, but also like an epigenome, and potentially can barcode other things as well directly in the tissue. But afterwards, you perform your downstream chemistry in the tissue. Tissue itself is in a reaction chamber to do all the fascinating chemistry to do to achieve spatial mapping. So the paper we published 2020 reported how this in-tissue barcoding worked out. We used microfluidics to deliver the barcodes, but there are potentially many other ways to deliver the spatial barcodes into the tissue matrix. And also since then, I think quite a few laboratories around the world were successfully established the technology in their own laboratory. For example, two groups there, and one from Munich, Matthias Meyer, was totally kind of amazed that they were not only able to kind of establish the technology in their own lab, they were able to do much better. So now we are using their protocol. They can get a much higher gene count and a UMI count. But also, at every experiment, they were able to map out 9 tissue sections simultaneously by redesigning the whole microfluidic network. And a couple of other groups kind of tweak our microfluidic delivery system to create sort of the customer design DNA microarray, and you can have whatever microarray spot size you want, just using the microfluidic device, the microchannel to define. We tested this as well. Potentially, this can give you the size down to even 2 micrometers. So for delivering to tissue, that might be challenging, but for patterning on a glass surface, that's totally doable. All right, so how the technology works, this is our prototype device. This is the workflow. I try to keep it simple. On a high level, we use microfluidic device to deliver the first set of DNA barcodes, like A1 to A100. That's what we do now. And each of those, I think, no, this is still old protocol, and actually we're not doing this anymore. And the current protocol is we do bulk reverse transcription. So the tissue reverse transcription done in a bulk, you are no longer limited by diffusion reaction kinetics. If you're a chemical engineer, you know what I mean. But afterwards, we use the microfluidic device to just add ligation, the barcodes through 2 steps of ligation. So it turns out that temporal ligation is extremely efficient. The first ligation gives you barcodes. The first ligation gives you kind of X coordinates, and then the second set of barcodes gives you Y coordinates. When you ligate them together, you'll get the 2D grid of barcodes, A and B. So basically, each pixel has a unique combination of A and B. And afterwards, you collect all cDNA, which has been sort of spatially barcoded, okay, by those A and B. You'll deliver and ligate it into the tissue matrix, and you can do the parent sequencing to map out where those molecules are coming from, also to find out the identity of those molecules through your sequencing rates. All right, so recently we found, actually quite a number of years ago, we found this actually works on FFPE. This is just an overall approach is very versatile, and also in general we can get a much better sort of gene counts and a UMI counts. But FFPE sample is still challenging. But over the past 2 years, we really worked hard to make it so fully compatible with FFPE samples. Just to show you a couple of those examples, from some kind of easy ones like a mouse embryo to the challenging kind of clinical tissue specimens like a human lymph nodes lymphoma. And if you really compare the data quality, you can obtain using our in-tissue barcoding. And on FFPE, and so we were able to, for example, get more than 30,000 transcripts or UMIs per pixel. This experiment used the pixel size, which is equivalent to 10x Visium, to kind of have a fair comparison. And almost like 7,000 genes per SPA or per pixel. We compare this across different samples and also compare to the probe-based 10x Visium, which is not a base-by-base sequencing, but in our case, this is a base-by-base sequencing. I was totally blown away. If you look at the performance, we were able to detect even more genes per SPA as compared to the probe-based spatial transcriptome sequencing. Okay, after we published our work back to 2020, so a couple months later, Nature Methods chose the spatial transcriptomics as the method year. I know everyone in our community got so excited about this. That's a real recognition of the major leap to develop the spatial transcriptome sequencing, but also recognize the far-reaching impact this type of technology going to give you in almost every corner of the biological research. So in the same issue, they also highlight our work saying the multi-omics sequencing goes spatial because in that cell paper, we demonstrate not just a whole transcriptome, but also a panel of like 22 to 32 proteins. So back to that time, people thought that's pretty impressive, okay? But I felt that's still a little bit cheating to call that proteome. And then I asked my student, can we really do hundreds of proteins, potentially thousands of those? I would say for academic laboratories, that's probably a little bit overstretched if you really want to do thousands just to build that antibody panel. But let's just try hundreds. That's within the range we're able to manage. It's still challenging. And so we demonstrate it works quite well for both human and mouse tissue and a variety of human and mouse tissue types. As shown on the right, is a variety of different mouse tissue types. And I just show you one more examples. And that's very challenging tissue specimens, human heart valve. That's a mitral valve. We also have data from aortic valve. This valve does not have a lot of cells and a relatively low sort of gene counts, UMI counts, but full of extracellular matrix. And when you do a single cell sequencing, you never get an extracellular matrix protein into your sequencing data, right? We often call that the dark matter of your single cell sequencing data set. But in spatial, yeah, actually you can map down where different extracellular matrix protein are located. So in this spatial size-6 sort of spatial protein transcriptome co-profiling, we also include a panel of 16 ECM molecule specific DNA but conjugates to simultaneously map down not just the whole transcriptome and a whole kind of 300 proteins, but we also include a panel of 16 extracellular matrix proteins. Now you can really see where they are, although there's now kind of subcellular or single cell resolution, but you can definitely recognize the different regions and the different ECM molecules are localizing. And the unsupervised the clustering based on proteins, based on transcriptome, you get a more or less identical clusters, which is great. It's a good validation. And then the power of the high-plex proteomics. When we used very small panel, unless you carefully pick the proteins you want to put into that panel, we found it is not possible to use the proteins alone to do unsupervised clustering. But once you get a 300, you get a somewhat kind of unbiased global analysis. What I want to point out, even in this case, we use only 10% of the sequencing length to map down 300 proteins. In theory, all right, if you have like a 3,000 DNA antibody conjugates, that's durable. Yeah, potentially can map down all 3,000 proteins to really achieve kind of proteome-wide spatial protein expression analysis. So in my laboratory, we're always interested in more kind of mechanism, what drives the gene expression, what determines the expression of those genes, rather than, okay, what genes are expressed. Even in the whole spatial transcriptome field, after many years, I would say that's equivalent to spatial gene expression, right? So you are not looking that much beyond gene expression, but actually RNA biology is very rich. I think one paper I comment on, published in Science from Xiao Wang's lab at Broad, she was able to map down or image the translating messenger RNAs. I think that's a perfect example. And the RNA biology is very rich. Now we just look at gene expression. We just look at the presence of the messenger RNAs. We actually miss a lot of cool, exciting biology. And for my laboratory, we start to think about some the underlying mechanism driving gene expression or determining gene expression and epigenetic regulation. In epigenetic regulation, there are many different epigenetic regulation mechanisms out there. We were interested in DNA methylation very early on, but we decided to work on this too first. So of global accessibility of chromatin and also the specific histone modifications. So the global chromatin accessibility analysis, the very elegant chemistry developed by Will Greenleaf and Howard Chang, and also Jason here. Jason, the first paper, first author of that paper, reporting the chemistry called ataxic. So what ataxic works does is you isolate nuclei. Okay, that's the first step, and then use the TM5 transposase to treat the permeabilized nuclei, and the TM5 transposase has been pre-assembled with DNA oligo. You know the sequence of those DNA oligos. What this enzyme does is the enzyme cleave the genomic DNA and insert those DNA oligos into the cleaved genomic DNA loci. So where they're going to cleave, I think if they're wrapped around by histone, and that's inaccessible. If that's open, that's accessible, that can be cleaved. So basically, you look at all the cleavable sites of those genomic DNA, you can map down the open chromatin state or chromatin accessibility profile. But it turns out, if you do ataxic, I think everyone will tell you, try to get a high quality nuclei, and that's very important. That's the first step. You don't get a high quality nuclear extraction, you just don't get your experiment work. We found that's actually now so necessary. You can just get a thin tissue section, the direct process the thin tissue section with the TM5 oligo construct, and you can insert those DNA oligos into the nuclear genomic DNA direct in situ in the tissue. But in our case, we did 2 more steps to add a spatial barcodes A and a spatial barcodes B. So now, after you sequence, you get basically a pixel by pixel, kind of like every experiment you do. Every time you get thousands of ataxic experiments done, and every experiment corresponds to one of the specific pixels in the whole tissue. And if you aggregate some of those pixels, you can produce called a pseudo bulk. For example, we aggregate all the pixels in embryonic mouse liver. You'll generate this pseudo bulk embryonic mouse liver atac trace, and you see all the peaks, the location of the peaks in relation to the gene promoter, gene body, and now you really see all the kind of gene regulation, gene regulatory network stuff you are able to map out. So we collected the data from mouse embryo, I believe it's E12, and on the pixels you can sort of cluster them very much like how you do a single cell ataxic. But afterwards, you'll get those clusters. You can kind of put those clusters back to the tissue and see where they are. Okay, that's the power of the spatial omics sequencing data. And so, for example, right here, this cluster, you see, oh wow, this is actually liver, pretty much this exclusively in liver. And in brain, you see many different clusters already. So that's at this particular stage, I think a very diverse neuronal or brain tissue types already started to develop. You can look at those individual genes, but what you are looking at is now gene expression. Just to keep in mind, what you are looking at is This is gene activity epigenetic state. This particular gene is associated with erythropoiesis or red blood cells. You see this exclusively in liver, because at this particular stage, liver is the organ producing red blood cells, not bone marrow. You can look at many other genes. I think those, in particular, those ones strongly associated with the neuronal tissue development. Now you can see how those genes started to open, or the locus started to open. You can directly visualize that in the tissue. We also benchmark our data against the standard ENCODE reference data, I believe from Professor Bin Zhen's lab. You can see the co-embedding clearly tells you strong concordance, like a limb versus limb, but we see much more granular data points, because our data is kind of pixel by pixel mapping across the entire organ, and this is sort of like a micro dissected tissue to perform ataxic. What is much more powerful is you can integrate the single cell sequencing data and the spatial. That's what we always feel this is very, very powerful. The single cell sequencing data, if that has been well annotated, you have all the cell types identified, that's terrific. And when you co-embed the single cell and the spatial transcript, spatial attack, in this case, spatial epigenome data, you can see where the different pixels showing in the black dots, so co-localized with which cell cluster or which cell type in this co-embedded UMAP. So that's kind of simple approach to find out where what major cell types are in which pixel. We're not computational biology lab. Actually, the computational folks can do even better job. They can resolve not just the major cell types in each spot, they can also further decompose or deconvolute each spot to identify even some minor cell subsets or a mixture of different subsets. But we try to just figure out the major cell types in every single pixel and then go back to see where the different cells are in the tissue. So just to show you a couple of those, and for example, the urethral lineage, again, that's in liver, and you'll see the different neuronal cell types, etc. neurons, and some post-mitotic premature neurons, and some oligodendrocyte lineage. So now you can really see where they are in terms of their epigenetic state. So that's important. You are not just seeing the gene expression. You know that very early on, the chromatin started to open, and the cells probably, that's the very beginning, they started to make a commitment to that particular lineage. All right, so in my laboratory over the past year or a half, we got very excited about the other side of the story. So we look at the embryo development, but now we're thinking aging, and turns out senescence is a very important mechanism associated with aging, but I don't want to say senescence equal to aging, or aging always just driven by senescence. Turns out senescence is a very interesting, very important mechanism modulating physiological function. We're interested in immunosenescence, and or broadly speaking, senescence in the lymph organs and also immune cells. The hallmarks people identify and also a whole set of genes. And so now we're able to look at them one by one in our kind of spatial sequencing data. For example, this is data we collected recently, still not perfect yet. It's still ongoing work, but pretty exciting. This is, I believe this is liver, and this is a human liver. So using spatial ataxic, you are able to clearly resolve those kind of different zones. We know the liver zonation from portal vein to central vein, so zone 1, 2, and 3. We're able to clearly resolve those based on the attack chromatin accessibility profile from all the pixels. Since this is genome-wide data, all right, so you can always look at those senescence markers, senescence signature genes, for example, P16, P21, and gamma H2 and HMGB1. So what we observe is those senescence markers are clearly kind of zonation specific, at least at this stage in the normal aging human liver. And they don't always co-localize, or they don't always overlap, indicating you can call them senescent cells or senescent-like cells, but they are not just one type of cell. They are very heterogeneous. I still don't have the full story to tell, but my intuitive hypothesis is during this kind of normal physiological aging process, we continuously accumulate those senescence-like cells through very distinct, very different pathways. And those 4 markers we choose represent 4 different biological pathways for the cells to become senescent or senescent-like. They can go through all different pathways, but once they converge, all those markers started to express, that's the moment those cells cannot revert back to non-senescent or cannot be clear or difficult to be clear by the immune cells. That's the moment I think the cells started to make trouble. But how they make trouble? They are rare. If they are rare, if they don't do anything, we don't care, right? So the problem is those cells gonna produce a lot of secreted factors, those SASP factors messing up the whole tissue microenvironment. So that's also something we can find out using the spatial data. Single cell data, very challenging to do, but spatial data, you can directly visualize those converged senescent cells and see how the surrounding cells become different once the senescent cells or senescent-like cells present there. I'm not showing that result, but we have some data to kind of point towards that direction, but too early to make that conclusion. I would say that's still my hypothetic thinking at this moment. So beyond global chromatin accessibility analysis, we're able to look at specific histone modification as well. And so in this case, we look at 3 different histone marks. As long as you have good antibodies, I'm sure you can apply this technology to look at whatever histone modifications. And 3 histone mark, H3, K27 trimethyl, K4 trimethyl, and K27 acetylation. I think 2 of those are kind of enhancer promoter, kind of promoter activity mark, and the other enhancer promoter, and the other one is suppressive mark. And from either one, you can perform this unsupervised clustering to see where the different tissue types started to develop at this stage, mediate or regulated by this histone mark. Okay, that's the key message. You are not trying to just identify cell types or tissue type. People constantly ask me this question. I did, okay, this may not be a valid question. Which sort of data can better identify cell type. In this case, the goal is not to identify cell types, but actually identify or reveal the mechanism whether or not that histone modification does play a role in the development of that particular tissue type in this whole organism. And so what we found is, okay, from either modality, you can pretty much resolve major tissue type, that's true, but you still see some difference, right? For example, right here, you can resolve many different tissue features, different tissue types, but this histone mark does not, I think, in this particular region, but maybe other regions. So now you get the idea. So when you do ChIP-seq, you do multiplex, you can put multiple antibodies there to do ChIP-seq, and that's the way to resolve how different histone mark kind of work together, such as bivalence effect, right? But in this case, even though without doing that multiplex cut and tag, you are able to see some sort of multivalence effect or collective effect by the different histone marks by comparing the data across several tissue sections. I feel that's very fascinating. So again, the benchmark against the ANCO reference data gives you the confidence it's a pretty strong concordance. And you can look at on pixels after clustering, you put it back to the tissue and see where they are. And it's pretty impressive. You can see how fine you can resolve the different tissue features even in this brain kind of forebrain region. It's a very sharp interface you can resolve using the spatial epigenome profiling. That's what we observe very often. So it seems like in the epigenome data, you see extremely sharp separation between the tissue types, even very, very subtle difference you can see from the transcriptome, but you see that a sharp change in the epigenetic state. So you can, again, this is genome-wide sequencing. You can map out on the peaks where they are, and you can visualize the location of the peaks in relation to individual genes to look into the promoter activity and the peak interaction and so on and so forth. We haven't done that much, but the data extremely rich. Actually, if you're interested, you can look into a lot of kind of mechanistic epigenetic regulations. After you look at those genes, you can also go back to visualize where they are. Just show you a couple of those. But keep in mind, this is now spatial gene expression map. This is also not spatial accessibility map. This tells you, all right, whether or not that particular gene in this particular organ, for example, liver, is associated with this specific histone modification, this particular epigenetic mechanism. So altogether, what you learn here is the epigenetic mechanism. All right. So when you have the single cell data, we also have a single cell content tag. I think we include in this paper. But what I want to show is, even if you don't have, it turns out this co-embedding with single cell transcriptome sequencing data also worked out so well. And to some degree, I found that's complementary. That gives you even better results. And so when you have the single cell whole transcriptome sequencing data from this mouse embryo, and we co-embed with our single cell content tag, you can use this label transfer to identify all different cell types and looking for the overlapping region in this spatial content tag, and go back to see where the different cells are. I just show you a couple of those. So recently, I think since we were able to do spatially resolved epigenome profiling, epigenome sequencing, and a whole transcriptome, and the reason to do spatial epigenome profiling is we know, okay, that's the mechanism potentially controlling the expression of the gene, right? And then you may ask, how about you actually measure an expression of that gene and just really compare? So that's going to be a home run to finish this story. And so we found that's actually totally durable. That's not too challenging for us to do, but I don't want to, kind of underestimate the amount of work my postdoc did. So in theory, pretty straightforward, but still a lot of optimization because the two assays are not, kind of naturally compatible. So a lot of kind of tweak and optimization, in particular from the tissue polymerization and the fixing, we need to optimize to make it work for both modalities. What I'm showing here is just one representative data from all three histone marks, and from every single sample we're able to map out the spatial transcriptome. From either transcriptome or epigenome, you see, we can pretty much resolve all major tissue features in this mouse brain. It's almost half of the juvenile mouse brain. And so you can sort of get all the major tissue types. And now the question, I think one thing is, okay, people also constantly ask, when you do spatial transcriptome, whether or not you need to replicate. My answer is often I don't want to do replicates, or that's not so necessary. I think a large-scale, big omics data. Instead, if you have the budget to do multiple samples, I would say do multiple samples over different times, or kind of set up biologically meaningful samples to look at. But still, if you can do replicate, you see highly consistent spatial patterns. That's still cool. And in this experiment, we didn't do replicates, but from every sample, we have the spatial transcriptome. This kind of like a replicates, and you see how consistent the different tissue features identify from all the three different mouse brains. But again, once you have maybe I go to next slide. Once you have the spatial transcriptome and a spatial epigenome. And as I said, either modality can resolve all major tissue types. I remember a while ago when I kind of talked to Rahul Satya, and he also mentioned that no matter single cell ATAC or single cell transcriptome, you can resolve all major cell types. There's no that much added value. But I would say, because you are trying to just identify major cell types, right? Major cell types fully determined by either accessibility or gene expression, that's not a question. But you can either identify in a rare state, okay, at the very beginning of the epigenetic turn on and the gene is still not expressed, can really look into that difference. That difference is where the biology is more exciting. So we probably skip those, because I feel this is a pretty exciting result. So now once you have both modalities, I want to kind of challenge my postdoc. What's the added value once you have both, right? And she performed this kind of pseudo time to look into the radioglia differentiation to postmitotic premature neurons, and also I remember when we submitted the first manuscript, we were looking into all the downstream lineages, I think exterior neurons, inhibitory neurons, and interneurons. Very fascinating cell differentiation trajectories like a spider. And I was so kind of impressed with that result, but a lot of pushback from reviewers. It was very difficult to understand. So, okay, that's fine. Let's just restrict this to the very straightforward radioglia to very early stage premature postmitotic neurons. And within this kind of differentiation process, you can map out on a pixel sensor where they are. I think the radioglia cells localized in the ventricle, once they are committed to neuronal lineage, they migrate towards the brain. And so that's exactly what we saw from our spatial data. And also from the only pixels, you can perform this pseudo time to look at the genes associated with this differentiation process. Now you can look at that pseudo time from two different aspects, okay, either epigenetic or transcriptome. So now when you put them together, you found actually pretty interesting. They're not identical, right? Or maybe most of them are following the same trend, but you do see, at least in the ones I show you, they're not identical. In general, and that still follows our hypothesis. And the chromatins started to open, and then later the genes become expressed. Okay, that's kind of what you expected. I think many of those kind of follow this trend. But we saw a group of those, in particular right here. And so the chromatin started to open at this time. But those genes remain open in terms of chromatin state. But the transcripts actually expressed very early on and quickly diminish. So the transcripts for those genes no longer expressed, but the chromatin remain open. And when we look at those genes, my collaborator Gonzalo Castillo Branco got so excited about, because those are his favorite genes associated with oligodendrocytes. They're not for neurons. And the implication here is, okay, even though those cells already decided I'm going to be neurons, okay, I'm going to become neuronal lineage cells, but they somehow still retain their chromatin state for other lineages, in particular in this case, oligodendrocyte lineages. And during the developmental stage, we didn't see that they were able to transdifferentiate into oligodendrocyte, but something goes wrong. All right, he found, okay, in the neuroinflammation, some of those premature neurons might transdifferentiate into oligodendrocyte, and some of those oligodendrocytes are even very distinct phenotype. They're called immune oligodendrocyte that really behave like inflammatory cells driving the pathogenesis or play a role in the pathogenesis. I think that makes the story all of a sudden very clear and very complete. All right, so as I said, we're also able to do more kind of trajectory, differentiation trajectory analysis. So we try to ask, okay, some of those cells still remain the potential for oligodendrocyte lineages. And when we use the spatial ataxic data and a spatial transcriptome, the RNA data to perform this lineage analysis, this differentiating lineage analysis, now you do see, okay, indeed, the epigenome data, you see this bifurcation, but for transcriptome data now really there, they still follow this linear differentiation. The cells behave like neuronal lineage, okay, that's not a problem. But if you look at the epigenetics data, you see this bifurcation, and if you go into the spatial, you do see the green and the blue dots, the different cells, they're not spatially separated, they are actually kind of mingled together in this brain region, but they are epigenetically different. One subset has much higher potential to transdifferentiate into oligodendrocyte lineages. You can look at different genes associated with the two different, we call different states. This green and the blue, even though they already told themselves, I'm going to become neuron, but they still have two slightly different epigenetic states. All right, so recently, so we say, okay, at least in some cases, right, so for example, neuroinflammation, you want to look at a lot of immune genes and the surface markers associated with different immune cell types. Can we get all them together, sort of the protein, transcriptome, and the epigenome? I just want to show you that's durable, and actually they work out quite well. But we were working on this for brain samples, and most of those gene markers we demonstrate in the Nature Biotech papers are just not so relevant. We include a couple of more markers specific to brain. This is one of those you can see from the protein data and from the epigenome and the transcriptome. You do see very consistent results. So now what is more interesting is now we apply the spatial triomic sequencing to the neuroinflammation study. This is mouse model developed by Gonzalo's lab to mimic the MS, like a multiple sclerosis, to see how the neuroinflammation drives the demyelination and oligodendrocyte damage, demyelination, and eventually neuronal death, neuronal damage. So now we're able to map down the whole process across all three layers, the epigenome, transcriptome, and in this case, over 100 proteins. So just a little bit about what we believe, I think the real power to go beyond what we do in my laboratory is sequencing. So I love imaging, just as I said, I think that's one of the two major categories. I believe in the real powers, we can combine imaging and spatial transcriptome or spatial multi-omics. And this is a beautiful work done by former postdoc now, former graduate student now postdoc at Columbia. They mapped out human brain hippocampal region from, so now I think the data is updated now. This is 16 human brains. I believe 6 of those are from very kind of saddening stories. There's a major depressive disease patients that committed suicide, and then we were able to get their brain and look into their hippocampal difference. And so for each sample, we were able to finish in the single cell spatial transcriptome, spatial attack, but also we did a high-plex immunofluorescence. We developed our own panel because the commercialized panel are not really compatible with human brain cell types. We developed this panel to really map out all major cell types in brain. And from the raw image and the cell segmentation, you'll get all the different cell types. It's hard to see. This is huge data. But if you zoom in, you see this is individual dots. This is individual dots correspond to individual cells. So also we can take all those segmented cells to perform this clustering and show the in the UMAP. Then you know what type of cells, but you definitely can very much like our spatial transcriptome data. You can go back to see where the different cells are. Very interesting. I think most data sets people collected, they never cover this region. There's a choroplax. That's basically a very small piece of tissue we cover in this hippocampal region, but that's not hippocampus. That's the kind of cerebral spinal fluid producing epithelial tissue in brain. All right, so now, as I said, the same tissue serial sections were able to generate spatial transcriptome, showing on the top panel, spatial epigenome data. Now we try to integrate everything together. I believe that gonna give you a much more complete story. Anything else we should incorporate? I believe the functions, right? So when we look at a protein gene expression and epigenetic, that tells you the mechanism about what's the real function. Proteins do a lot of functions, but another important aspect is the metabolism. Right, so recently we collaborate with Professor Lin Yanxi at UCSD to combine this kind of metabolic imaging using stimulated Raman microscopy and the codex. We did a codex first, but potentially we can combine this with spatial transcriptome epigenome sequencing as well. What is cool about this approach is completely label-free, so you don't really damage the tissue at all. And after this metabolic imaging, you can take the same tissue to perform, in this case, the high-plex immunofluorescence imaging, or you can also do spatial epigenome transcriptome and then map every cell you see here to their epigenetic, to their metabolic state. For example, on the lipid metabolism, you can actually see lipid droplets, which is now easy to do using mass spec. I think the optical microscopy approach allows you to see individual lipid droplets, lipid dynamics. And also you can look into something like a glycolysis versus OXPHOS. We're interested in T-cell biology, T-cell, B-cell biology, that glycolysis versus OXPHOS is very important. Now you can see that important metabolic function cell by cell and link that directly to, for example, the codex data. So just to very quickly summarize, I don't think I really need to summarize. Just 2 sentences. Okay, we developed a bunch of tools, but now for fun, okay, for some real applications. I think the important applications, I would say almost everything, almost every corner of the biological and biomedical research. But we have small group. We focus on 2 things. Okay, one is I think the senescence, aging, and immunobiology, and when you combine, that's really kind of my passion. So immune system, what they do, I think the different immune cells in disease and also in aging. We're also part of the Brain Initiative working on brain cell atlas mapping and to really understand how we think. Okay, I think these 2 things are probably most important to my opinion, how our immune system works and how our brain works. All right. I would like to acknowledge you folks in my laboratory and also all the amazing collaborators around the world and also my funding support. And thank you so much for your attention. And if time permits, I'm happy to answer a couple of questions.
One in front. There we go. Thanks. Reminder, folks, please do use the microphones asking questions. Thank you. That was amazing. How, especially now that it is possible to do so many omics in a piece of tissue, how does a researcher decide what is the right number of omics for them to do? Is it just literally everything you can afford, or are there cases where you think some of these add particular extra value? How do you decide what to do?
Yeah. Okay, maybe the question can be kind of broken down into two different things. One is so many different technologies or modalities out there, right? So you can kind of use codex and use. So which one should you choose? Everything is expensive. So I would say it depends on your question. So I tend to get a complementary data. But sometimes you may say, okay, I want to get an identical data type, which means, okay, gene expression, then you want a fish, you want a spatial transcriptome. But I tend to like complementary data that can give you a more complete picture. But on the flip side is, oftentimes you can now just directly integrate them together very well and a lot more computational. So it depends on what, how deep you want to go to the computational to really kind of either kind of disentangle or find the links between so distinct data sets to make sense of the biology. And the second is, so different modalities, different omics, can you do all of them on the same tissue? Because that really gives you sort of the ground truth, right, on the machine learning AI stuff. But they can sort of use label transfer, project this type of data to another. But what's the ground truth? Ground truth is you actually have data from both on the exact same cell in the same tissue. So that's what we try to push in my laboratory. But once we get that down, maybe one day you don't have to do that. On the AI machine learning getting more and more powerful, in particular when the data become available to train those AI models. Also on the other hand, I think for this multi-omics, the same tissue, same cell, one challenge is, okay, every experiment has some failure rate, right? If that failure rate is 10%, which is pretty good, it's, if the 30% actually acceptable in academic laboratories, but if you combine 3 modalities on the same tissue, you can do your own math. So literally, the student gonna be very frustrated. So just think about all those. I feel that's probably my practical thinking. Number one, the biology, the question you wanna ask. Number 2, how feasible, you don't want to frustrate your students. Pretty much those 2 things. And I didn't talk about budget because I know many of you guys are from the industry, you don't worry about the budget.
I think this was fascinating. So coming from the more drug discovery pharmacology angle, I was wondering if you have thought about, kind of shedding light into the black box of what happens when a molecule works in an animal model, or maybe it does not work. So for example, some of your multiple sclerosis endpoints, right? It would be very cool to see what happens to, those profiles when, those animals have been subjected to a small molecule that is going or is already in clinical trials, to see how that actually affects the whole composition of the tissue. Because, we know that molecules act at many targets in addition to its main target. And I wonder that would be an interesting layer to add to the, to our understanding of the pharmacology.
Okay, maybe I want to kind of make sure I understand the question. You're talking about the molecules in the drugs?
Yeah, so when we in drug discovery perform our studies, we typically have a molecule that starts with a cell-based assay. We progress it all the way to an animal model. We say it works here, let's go and put it into humans. But it's very little information we really have from those brains many times.
From those animals.
But we know that molecules, over extended time, they will actually bind to many things, and they will do a lot of things to the biology of the tissue. So I wonder if your approaches would be able to shed some light into some of those kind of, black boxes that we have in the industry.
Yeah, I now totally understand your question. That's we're on the same line. But I don't want to kind of overclaim too much at this stage, and because I think most of my amazing colleagues in medical school, they love animals, they have animal models. But my personal opinion, I think the most informative and the most kind of truthful experiment is clinical trial, it's the human data, right? So I constantly emphasize, can we really understand human biology, all those kind of pharmacological clinical studies? But indeed, unfortunately, most assays are now kind of fully compatible with clinical specimen, but now we are getting there. So I really hope we can apply those tools, including what I talk about is high quality spatial FFP RNA profiling to all the clinical tissue specimens to really uncover the mechanism derived from clinical trials. That's what I hope to see. But animal models still indispensable, and if you are in Europe, you may argue the organoids are becoming indispensable. Yeah, so that's where we are, but we are unlocking tremendous new opportunities to really look into human biology or human disease biology in humans.
Thank you so much. That was really incredible.
Thank you very much.